Hi, I'm Yossi Farjoun. I'm a DSP as well in the methods, like Mark. And this is going to be just a brief introduction to the data preprocessing that we do on the genetic data, mapping and cleaning up the sequence data. So Mark talked a little bit about how we take the sample and we sequence it, and we get reads, and we want to talk about what happens in the first step of these reads, how they come off the sequencer, and what are the first few things we do to them before we think about variants. Just how do we get the reads to a state that we can even talk about looking for variants. So basically, we're going to be doing three things. We're going to map them or align them to a reference. We are going to mark duplicates, which connects to the question that was just asked. So how do we know whether we are sequencing the same molecule over and over again, or if we're getting new molecules? And we're going to do base recalibration, which enables us to get a better estimate on the quality scores of each of the bases. Feel free to stop me if you have any questions.
So it may calculate coverage. The duplicate doesn't count, right? If the reads come from one molecule, it's only count one, right?
Right. So let me repeat the question. The question is, when we estimate depth or coverage, do the duplicates count? So it really depends on how you run the tools, but most tools' defaults are so that they ignore duplicates. So if you have marked duplicates, if you ran marked duplicates and you've marked duplicates, then the downstream tools, unless you try hard, they will not count duplicates. All right, so here's the data processing kind of workflow that we have. We start with unmapped reads, raw unmapped reads. They can either come in a fast queue or in an unaligned BAM file. We'll talk about what those are. We map them to a reference. So now we have mapped reads. Before we had unmapped reads. Then we mark duplicates and we recalibrate the quality scores. And we have analysis-ready reads. Right? So these are ready for, if you're doing variant calling, they're ready for variant calling. If you're doing RNA expression counting, they're ready for that. Right, and as a reminder, when the data comes off the sequencer, it's a pile of reads that has no mapping information. It's sorted by, it's a physical sorting. It's sorted by the position on the sequencer, basically. And it has, technical artifacts and biases. It has duplicates. It might have library duplicates or sequencing duplicates. We can distinguish between them, but we don't really care, usually, unless we're in the lab and we want to minimize one or the other. But as an analyst, we usually don't care what kind of duplicate it is. All right, and so we need to, as I said, map, sort, and clean up and fix the quality scores. So first thing is align. Basically, an alignment is looking at a read, each read or each read pair, and finding the best place in the reference where that read fits. And not only where it fits, but specifically how it fits. If there are specific insertions or deletions, some alignment information that needs to be adjusted between the read and the reference locally, the alignment does that as well. Right? So it tells us the read is over here in the reference, and also, by the way, you need to have this insertion of a few bases over here and a deletion of a few bases over there, and then this is the best fit. Right? So that's what we call alignment. We use BWA MEM for DNA. We use STAR, 2-pass STAR for RNA-seq. That's our current recommended practice. And so if you start with a pile of reads that the colors here the colors are visual representation of the same place in the genome. Right? So you can see that originally they're not aligned. Once you align them, then you can put them so that they look like what you expect in IGV, where reads that came from similar places in the genome are positioned in the same place in the file, and they have their position information. Okay? So that's what the aligner does. So as a caricature, here is what we are looking for. So we have a reference that has several regions. Our sample is different from the reference, right? It could have local differences, like these stars are just maybe SNVs or indels, or it could have large differences. For example, this region is a copy number duplication, right? So it's a large region that's been duplicated relative to the reference. And so when we sequence that, we might have some confusing, we might have some artifacts that, or not artifacts, we might have some results that look a bit confusing because we'll have reads that come from here and reads that come from here, all aligned to the same place. Okay. Whoops. That's not the right button. There we go. Yes?
How does it know if it's a copy number variation or if it's duplicated?
The aligner doesn't know. The aligner has no idea. If the reads are shorter than the duplicated region, then all the reads over here will go here, and all the reads over here will also go here. And so that's why we need, and even the kind of first attempt tools that we have to have caller and mutect and what have you, they will not know either. We might have some filters that will be able to sniff out that something is weird. The allele balances are off. There's all kind of strange results. There's mapping. There's things that look like mapping artifacts. There's certain smells of this thing happening. But in order to absolutely know what's going on, you need to run a copy number. You need to run a copy number tool. We have a copy number suite of tools that you can run, and it will tell you, oh, there's a copy number here. And then we actually don't have yet a good answer for what do you do next. How do you find out, which copy had these variants and which copy had those variants? We haven't developed. I'm not aware of anyone that has developed tools that will actually open this up from short reads. From long reads, it's easy, right? You read the whole thing through and you're done. But with short reads, it's hard.
But if you did get a read spanning the two regions, the aligner still can't say it's an additional copy?
The aligner will not say. The aligner's job is to tell you where in the reference this read goes. That's it. It does not, it's unable to say, oh, this looks like an extra copy. What it will say when it looks at a read over here that kind of spans over here, it will say, this is very confusing. It looks like it comes from here, but then it doesn't match over there. And so it clips off the end, or it does all kinds of strange things. So those artifacts will be visible in your file. You will see that at some point there's a whole list of reads that are clipped. And so that's an indication that there's a problem. But it could also be an indication that there's some bacterial contamination. So, you have to, it's not so simple. Okay, so in the easy case, when we just have the reads, there's no replication, everything is different from everything else, then the aligner has an easy job. But sometimes it's not so simple. Sometimes the reference itself has replicated regions, right? So maybe it's some ALU or some other biological thing or who knows what. The reference is what it is. We don't control it. So if we have if we have reads that come from here, the aligner has to decide where they go, even though they align well to both places. And so in that case, we'll end up, there's two things. The aligner will try to use some tricks with a paired end. So for example, here, there's a replicate, but the pair helps you. You kind of know what the insert size distribution is, and so you can match the location that fits better given the position of the mate. But of course, some cases, even the mate is included in the replicate region. You got a replicate region that's 2,000 bases long, and the mate's not going to help you there. And so then you're going to get low mapping quality, and that's, if you see a low mapping quality region, that's why. Okay, so what is the output of the aligner? It's a SAM or BAM file. You can look at these files using SAM tools, for example. And the reads, the file is organized in the following way. We have a header, which tells us some information about the overall structure of the file. So we have the version, we have whether it's sorted by coordinate or other, we can sort it by different things. What the sequence that was used in the reference, so it's kind of a dictionary of the sequences that were used, and then what read groups are included in this file. And then after the header, there are records that all look like this, and they have information about the read and about the mate if the mate is present. So the name of the read, which is basically an encoding of the physical location where this read came from, the name of the machine, the full cell, the lane, et cetera, et cetera. Some flags that tell us bitwise information about the alignment of the read, whether it was aligned in the forward direction or in the backward direction, information about whether the mate was aligned in the forward direction or in the backward direction, and so on. The position and chromosome, or chromosome and position that it was aligned to, the mapping quality, a CIGAR, which I will get to, the position of the mate, the alignment position of the mate, the insert length, and then finally, the thing that we actually got out of the machine, which is the read sequence and the qualities. Right? So those are the original things that came out of the machine. Everything else was added by the aligner. And then the read group, if you have a read group, then this is where the read group information is going to go, and there's a lot of other tags that could end up being afterwards, certain tags that you can read the spec documents to your heart's content if you care about such things. Right. Okay, so a bit more information about the CIGAR. CIGAR is an acronym. It took me a long time to find that out. That's exactly what it stands for. Basically, a CIGAR is a concise way of writing down alignment information. Right? So, for example, here, this CIGAR says the first base is soft clipped. That's what the S stands for. Then we have 3 matches. So those are alignment matches, not base matches. Okay? So alignment matches means a base over here and a base over here, 3 bases. Right? Maybe the aligner decided to put a C against an A. That's the aligner's choice. It doesn't talk about SNPs, only talking about alignment information. Then there's a deletion of 1 base, then 2 more matching alignments, and then 1 insertion, and then 1 more match. That's what this is. Okay? So basically, if you try to draw this in your as a cartoon, this is where it is. This is the reference position. And here's the reference, and here's the read. And notice that even though, so here this is a soft clip, so it doesn't care what's going on here. These are matches, 3 matches. Then there's a deletion, 2 more matches, 1 insertion, and then 1 more match. Now, this A could have been anything, and it would still be 3M. Yes, question?
Good question. Why do we skip the first? So why is the first base?
The aligner chose to do that. The aligner chose to do that. So basically, the aligner is aware of the fact that reads, bases at the ends of the reads are suspect. And so it doesn't want to treat them necessarily as an insertion because that comes with a penalty. And so one way to get around that penalty is to say, okay, you're allowed to ignore bases at the ends of the reads. Only at the ends.
Why don't you call it a match? Because like can you get the first statement of the CIGAR string would be 4M.
So this is a cartoon. I'm not saying that this particular string of bases would get this particular alignment. Okay? And I don't want to give you an alignment of 120 bases because it will take all day to go through it, right? So, but the point is that the ends of the reads can be clipped. This is the moral of the story. It's not about this 1 base being clipped. Okay. So there's 2 standard ways of dealing with unmapped reads becoming mapped or aligned BAMs. One that most people are familiar is using FASTQ. So you have a reference genome, you have a FASTQ file, you pass it to BWA, you get your alignment information. The kind of twist here is that if you want read group information, that usually requires extra kind of manual intervention. Make sure you give BWA mem all the information about the read group and the sample, et cetera, so that it puts it in the header for you. We prefer not to modify the command line all the time. And so what we do is that we run BWA mem from a BAM. We get a mapped BAM that does not have any of the read group information. The read group information is stored in this unmapped BAM. And then we run a tool called merge BAM alignment that takes these two BAM files and effectively takes the alignment information from here and puts it onto the BAM that has all the read group information and all the metadata that we wanted to have. So this is our implementation detail using merge BAM alignment. It also does some extra cleanup. So for example, those bacterial reads that Mark showed in the previous talk, merge BAM alignment will identify them and unmap them for you. So it will clean it up a bit. Right? So things like that get cleaned up by merge BAM alignment. RNA needs slightly, in order to process RNA-seq data with GATK tools, we need to split reads that have a large N operator in their CIGAR. This N operator basically is different from an insertion because it doesn't carry the penalty of an insertion. Basically, it's saying there's an intron here, so it looks like a large insertion in the, a large deletion in the read, right? Because I effectively deleted the exon. As far as the read's concerned, the exon has been deleted, but I'm not going to call it a deletion. I'll call it an N, a skip, a gap. Okay? And gap-aware aligners, RNA aligners are aware of the exon structure, and so they know how to do this. But in order to deal with this with GATK, we need to do an extra step, which is not listed here. I guess we'll talk about that in a separate talk. But the point is that when you run RNA alignment, you get these gaps in your alignment. And so those gaps are like here, there's this N operator in the CIGAR. Question?
Yeah, so going back to the alignment then, if you had to make a choice between keeping FASTQ information or the alignment, then what would be your choice?
My choice would be using unmapped BAM. I don't like FASTQs. What ends up happening with FASTQs is that there's no natural way, there's no structured way of keeping metadata about the FASTQ. And so you end up having to store the metadata separately from the file, and I really don't like that, right? So you have one file containing metadata, and then the FASTQ file, and then you don't know which one is which, and you get a big mess, and I don't like that. I prefer all the metadata to be in one file with the reads and the information that's needed.
So this is probably a naive question. One thing I'm not quite grasping with the CIGAR, like you can have like 3 matched, and then you can have a deletion, 1 deletion, and then 2 matched. But your 3 matched don't actually have to be matching.
So it's not a. The important thing to remember about the match operator, the M operator in the CIGAR, is that it's an alignment match. So that if you were to put, like we did here, the reference and the read one above each other, alignment-wise they match. We go base by base. For every base in the reference, there is a base in the read. Unlike here, for example, where this is a deletion, there is a base in the reference and there is no base in the read.
But why did you decide to say that push that as a gap as opposed to just push over the CCAG line?
Because then you would get a lot of mismatches, right? So the job of the aligner is exactly to try to minimize the mismatches.
Okay. So it's looking first to find the best mismatches, and then after it does that, it assigns the CIGAR.
Correct. So there are aligners. I don't remember which one. Maybe Minimap. Anyway, there are aligners. So there are 2 more operators. There's a few more operators that I haven't talked about. But there are 2 operators that were defined that do take the bases into account. So there's an X and an equals. And so theoretically, X and equals are subclasses of M, right? That look at the base itself. But what ended up happening is that we don't use that information. We only care about whether it's an alignment match or whether it's an insertion or a deletion. All the downstream tools, they don't care so much about whether it's a X or an equals, whether the bases agree, because they will look at the bases. Sorry, it's not that they don't care. They don't look for this. They don't look to the CIGAR. They don't look to the CIGAR for that information. They will look that once they know that there's an alignment match, then you can just take the substrings in the reference and in the read, and you can compare them to yourself, and you can do all the work that you want. You don't need the CIGAR to tell you that. Does that make sense? Okay. All right. So going to step 2, so we have an alignment. Now we look at duplication. Duplication is a problem because in variant calling, we will count the same possible artifact multiple times if we have duplicates, right? So you could think of maybe there's a PCR artifact, and then it got read multiple times, and now it looks like you have 5 copies of the same artifact, and if you're a somatic caller or a germline caller, you might make a mistake. Another problem is in RNA, if you have duplicates and they're not real, then they could change the apparent expression levels of your different genes. And different genes could have different duplication rates for GC bias reasons or for all sorts of reasons, right? So it's not going to be a uniform effect on all of your genes. So that's why we remove or actually mark duplicates prior to analysis with a tool that's called mark duplicates. It's a very original name. And the important thing to know about the way mark duplicates work is that it does not at all look at the bases. Okay? It does not look at the bases. It looks at the alignments only. So the idea is that bases could be slightly different due to sequencing errors. We're not going to take a 150 base pair read compared to another 150 base pair read and declare them to be duplicates or not duplicates based on the specific bases that are in them. Because there could be a sequencing error. So then we're going to have to start having knobs and dials to say, oh, well, if it's just 2 reads or 2 bases that are different, we don't do that. We let the aligner decide. If it aligns in the same place, then it's a duplicate. If it does not align in the same place, then it's not a duplicate. That's basically the idea.
What about the soft clip? Does it show?
Good point. So we'll get to that in a second. So there are 2 kinds of duplicates that we are worried about. And this is where we know that they can come from. So one is PCR duplicates. I suspect everybody here knows what PCR duplicates are. So PCR is a process that we replicate the DNA. And so if we replicate our reads prior to reading them, replicates our library prior to reading it, we might get the same molecule read twice, right? So that makes sense. And the more cycles of PCR we have, the more likely it is that we will read duplicates. Another type of duplicate that could arise is from the sequencing itself. The sequencer creates these clusters of molecules that are supposed to arise from the same original molecule, and then it does this optical kind of recognition, trying to find what the different clusters are. And sometimes the clusters are oddly shaped, and it identifies the 2 clusters as one cluster as 2. Sometimes clusters are at the gap between 2 different images, and so the same cluster shows up twice, one in one image and one in the other image. Sometimes we have in the newer pattern flow cells, we have this jumping from one, because of the chemistry, we have this jumping effect where a molecule could spawn in one cell or one well, and then it could, some of its progeny can jump and kind of infest a neighboring well and produce another read. So all these things we know are happening. There's probably more that I haven't talked about, but these all are caught using mark duplicates. So how does mark duplicate work? I said we identify duplicates by position. I should have been more specific. We identify duplicates by the unclipped 5' end of the molecule, right? So we know that the read could be aligned forward strand or backward strand, right? We're going to look and say, where was the position of the first base that was sequenced, right? That's the 5' end of the molecule. And that's the position that we consider the key for mark duplicates. So whether that base was clipped or not clipped, whether that, we don't care for the actual position of the alignment position, which is always going to be the left side of the molecule. That's the alignment position. That's not what we look at. We look at the 5' end of the molecule, and we look at the 5' unclipped end of the molecule. And if it's paired, then we look at both 5' ends of the two reads. And the two reads, the read 1 and the read 2, and compare that to the both 5' and unclipped of the other two reads, right? And only in the case that all of these match, then we consider that a duplicate. Okay? So randomly matching your position without being a duplicate is not very likely for paired end. It's a little bit more likely for single-ended, right? So for single-ended reads, especially at high depth, you might get a lot of random, not artifactual duplication. And there you have to ask yourself the question, do I want to mark duplicates or not? We don't tend to deal with single-ended reads so much. Yes?
Do you only pay attention to the first base, or do you look as a k-mer? A k could be 5, so first 5, if they are the same, then it's a duplicate.
So like I said, we don't look at the bases at all. We ignore the bases. The bases are not considered at all for marking duplicates. Only the 5' end, the unclipped 5' end of the reads. The position in the reference of the unclipped 5' end of the molecule. That's the only thing that is looked at. Okay? So the only time that we do look inside we look at the qualities, or we look at the clipping in order to choose. So once we have a set of reads that we say, okay, these are all duplicates of each other, we need to choose one of them to be the representative, right? To be not marked as duplicate. And so for that, after we've identified the set of duplicate reads, we look inside the set and we choose the one that we think is the best. And there's various ways of choosing which one is the best. I'm not going to get into it. It's not that interesting. Definitely not going to get into it in this talk. But that's the only time. After we've identified the set, then we look inside to choose one of them. But for identifying it, we don't look at the bases at all.
So basically, you are stating that the event that you'll have two reads aligning with a starting alignment position at exactly the same place is very unlikely unless they are duplicated.
Exactly. Right. And we would rather remove the occasional randomly overlapping read than keep all of the artifactual overlapping read.
I have a question about this single-end read and paired-end reads.
Mm-hmm.
So I see some pipelines they do first merge the paired-end reads, then do the mapping. So
Yeah.
I don't do that.
Yeah.
So what's your suggestion?
So we don't deal with, so the question is that there are some pipelines that merge the first read and the second read into like a composite read. I am also familiar with these pipelines. It usually has to do with very short and poor quality reads. So for example, the ancient genomes pipelines do this. I'm not familiar with any other pipeline that's not part of the ancient genomes pipelines that does this, but I mean, I'm not familiar with all the pipelines. I question the importance of doing that. But I don't know enough detail, so I can't really comment. None of our pipelines do this. All right, so here's the effect of marking duplicates. So you can see here, if you look in IGV, and you can tell IGV to show the duplicates. So you can see that there's a lot of duplication, a lot of reads that look like they're duplicates here. And then when you don't show the duplicates, then there's fewer reads because they were marked. And so you asked IGV not to show you the duplicates, and then there's fewer. Make sense? Now, of course, when we mark paired ends, then just looking at one read, it's hard to see whether it's a duplicate or not. Right? So maybe this read and this read, they look the same, but without looking at the mate, you can't tell if they're actually duplicates or not. So if you want to kind of double check mark duplicates, you need to ask IGV to show you the reads as pairs, and then you can see, oh, okay, this one is not actually a duplicate because the other one is aligned differently. Does that make sense? All right, so the duplicates, oh, yes?
So when it's marking duplicates, it does consider both reads of the pair.
Correct.
If they're exactly identical by location, then it's duplicate.
Exactly, right. So the duplicate status is in one of those flags, the flags that I showed you before, right? So the flag, there's one of the bits says, yes, this is a duplicate. We don't remove, the standard way you run mark duplicates does not remove the duplicates, it just marks them as duplicates. You can ask mark duplicates to remove the duplicates for you. We don't do that. And then the downstream tools normally ignore duplicates. So without you having to do anything, most tools will ignore duplicates unless they don't need to or unless you tell them not to, right? So generally speaking, most tools downstreams are well behaved and they ignore the duplicates when they need to ignore them and they don't ignore them when they don't need to. All right. I don't know if I have, I'm running out of time. Local realignment has been deprecated and I don't want to spend that much time on it. Basically, this is a tool that was important for pileup based variant callers. So variant callers that don't know how to realign the reads, they just look at every locus and they say what bases are here, what bases are here, what bases are here on each pileup. And for those, you can have effects like this where the aligner is confused. You can see that the aligner put in things that look like SNPs. So if you were naive, you would say, oh, there's lots of heterozygote SNPs here, right? But if you realign these, when you have all this information together, then you realize actually all of this noise can be explained by one insertion. Sorry, one deletion in this position. And this deletion, because of the structure of the reference, only shows up as SNPs in the kind of down towards the edges of the tool. We no longer use indel realignment because we use haplotype caller, and haplotype caller does a local realignment before it looks at the reads, and so there's no need to do that. Yes?
But if you wanted to then visualize the reads for your own interest in like IGV or something, would this would be useful to make it easier?
If you wanted to, if you want, the haplotype caller talk, I don't know when it is, probably on day 2 or day 3, we'll talk about this. There's actually a flag for haplotype caller to give you the reads as it saw them after local realignment. It's called BAM out. And it can give you a BAM that not only tells you what the local realignment did, but also which read was assigned to which haplotype and a lot more information. And so if you want to do that, so for example, I don't know if you're familiar with the Nomad browser. So the Nomad project is this giant project we joined called, many tens of thousands of samples together, and we made a reference panel. And so you can click on some variants there, and it will show you screenshots of where the reads came from for those variants, and that's through this mechanism of BAM out, et cetera. So it's very convenient. Okay. Right. So as it says here, this is not necessary when using assembly-based variant callers like mutect and haplotype caller. Of course, cutting out. For RNA, you don't really care about the specific alignments. Usually you just look at your counting expressions, so it's not such a big deal. All right, so finally BQSR. We need to, the sequencer can only tell you how confident it is in the base relative to the base that came in to the sequencer. But if you put in an error in the PCR upstream of the sequencer will still tell you, hey, this is a really good base, right? And so if your PCR changes all the C's to G's, right? The sequencer will have no idea. It will say, yeah, there's a G, it's a really good G, everything's a G, right? But then if you look at the data, you'll say, this is really weird. All the C's turn to G's. I want any G base to come with a really low quality score because it's wrong, right? And so the nice thing about BQSR is that it looks at the whole file and it doesn't care where the error may have come from, whether it's sequencing or PCR or something else. The error will be recalculated, recalibrated for the entire sample, and the model is generated per sample. So we look at the sample, we compare it to places that we know there's no, or we expect there to be very little polymorphism in the reference. And in those sites, we just see what the quality is, we generate a model, and we apply it to all the bases. So you can see these are plots that show you before and after. So before, the quality score that was reported is not exactly the empirical quality score, but after we do the recalibration, then we get this nice straight line of blue bases. And then this is the same thing, but instead of segregated by quality, we segregate by the dinucleotide context, and we see again that we've corrected. There are some cases where, for example, here a CG dinucleotide context, there's a very big error between the reported and the actual quality score. But after passing it through BQSR, then that's fixed. So this is why we advocate for using BQSR. All right, this is basically what I said. This is what it does. So it changes the quality, and it copies, if you ask, it copies the original qualities to this extra tag in the read called OQ. We actually don't do that anymore in the pipeline because it basically doubles the size of the BAM. And, well, it's expensive. All right, so that's our introduction to the analysis. We start with raw reads. We map them, we mark duplicates, and then we fix the quality scores. If we use merge BAM alignment, we also clean up the bacterial contamination while we're doing that. And that's the end of the slideshow. Thank you very much.